You know, every time you sit down for a meal, you're looking at the end result of a story that's literally 10,000 years old. It started with the very first farmers just saving the best seeds. But that slow, patient process, well, it just hit hyperdrive. We're talking about tools that can predict a plant's future and even edit its DNA like a document. Okay, let's get into it. So here's the game plan for this explainer. We're going to start with the fundamental challenge that breeders have always faced. Then we'll dive into the genomic revolution, how we learn to predict a plant's potential. After that, it's all about the crazy precision of CRISPR, the time machine magic of speed breeding, and finally, we'll look at how all these tools are coming together to design the crops of the future. Right, so for pretty much all of human history, plant breeding has been about solving one single massive problem. A dilemma that has, honestly, shaped civilization as we know it. And that's the question, right? At its core, this is what drives all agricultural innovation. And let me tell you, the stakes today are higher than they have ever been. We have a global population that's just exploding, and at the same time, we're dealing with the massive pressures of climate change. 70%. Just let that number sink in. According to all the projections, we need to boost our agricultural output by a staggering 70% by 2050. Why? Just to feed the 9.6 billion people expected to be on this planet. This isn't just a small tweak, it's a monumental challenge. So if you look back, progress from most of history was, well, it was slow. It all started with just picking the best plants and saving their seeds. Simple enough. Then, boom, the 1800s, Gregor Mendel figures out the basic laws of inheritance, and suddenly breeding starts to look more like a science than an art. By the 1900s, we were doing controlled crosses, but it was still all about watching, waiting, and being patient. I mean, it could take decades to get one single new variety. And that's really the heart of the problem. Look, traditional methods got us incredibly far, but they had real limits. It's one thing to select for a simple trait, like making a seed bigger, but what about something like drought tolerance? That's not one gene. That's controlled by hundreds, maybe thousands of genes all working in a complex network. The old ways just couldn't handle that level of complexity. This exact challenge is what forced a huge shift in thinking. We had to go from just observing how a plant did in the field to actually predicting its performance based on its genetic code. And that, my friends, was the beginning of the genomic revolution. Now, the first baby steps in this direction involved tools like marker-assisted selection, or MAS. It let breeders track a handful of important genes, but for those really complex traits, it was still kind of like flying blind. Then came genomic selection, or GS, and this was a total game changer. Instead of a few genes, GS looks at thousands of genetic markers across the entire genome. It's like the difference between navigating a city with a few major landmarks versus using a live satellite GPS that sees every single street. So how on earth does this actually work? Let's break it down. First, you build what's called a training population. This is a huge, diverse group of plants. You grow them, you measure everything about them in the real world, their yield, how they handle stress, you name it, and you also sequence their entire genome. You feed all that data into a computer, which builds a predictive model. Now for the next batch of baby plants, the breeding population, all you have to do is read their DNA. The model can then predict, with scary accuracy, which ones are going to be the rock stars way before they even grow up. So to put it simply, it's a four-step loop. 1, you train the model with a ton of plants that have both genetic and real-world data. 2, you build the statistical model that connects the dots. 3, you take your new seedlings and just sequence their DNA. And 4, you use the model to pick the winners to be the parents for the next generation. You're basically skipping years of waiting and field work. Okay, so prediction is a massive leap forward. But what if? What if, instead of just selecting the best genes that are already there, you could go in and make precise, targeted improvements directly to a plant's DNA? Well, that brings us to the next huge breakthrough, the era of gene editing, and it's all powered by a little something called CRISPR. Honestly, the best way to think about CRISPR is like a find and replace function for biology. The system uses a guide molecule to search for a very specific sequence of DNA, and an enzyme, usually Cas9, acts like a pair of molecular scissors, making a perfect, clean cut right at that spot. It's so simple, and yet the power is just mind-boggling. We can now edit the source code of life. You know, CRISPR wasn't the first gene editing tool. There were others, but they were frankly a pain to work with. super expensive, took forever to design, and they weren't always reliable. CRISPR changed absolutely everything. It was simpler, it was way cheaper, and it was so ridiculously efficient that it basically democratized gene editing, putting this incredible power into the hands of scientists everywhere. So, you might be wondering, how do you get this CRISPR system inside a plant cell? Well, there are a couple of main ways. On the left, you've got a technique that uses a common soil bacteria called agrobacterium as a sort of natural delivery truck. And on the right, well, that's the gene gun. And yes, it does exactly what it sounds like. It literally shoots tiny gold particles coated with the CRISPR machinery right into the plant cells. Science is wild, right? And the potential here is just wow. With this kind of precision, breeders can go after almost any trait you can think of. We're talking about boosting crop yield, jacking up the nutritional value with more vitamins, tweaking flowering time to match the climate, even changing the physical shape of a plant to make it easier to harvest. And most importantly, we can build in powerful resistance to diseases and major stresses like drought and heat. All right, so we've got prediction and we've got precision. But there's still one huge bottleneck in breeding, and that's just time. A plant's life cycle is what it is, and that dictates the pace. But what if you could put that biological clock into overdrive? That's the whole idea behind speed breeding, and it's so brilliantly simple. You basically create the perfect endless summer for a plant. You blast it with customized light for up to 22 hours a day, keep the temperature just right, and basically trick it into growing and making seeds way, way faster than it normally would. Using this, breeders can now cycle through up to 6 generations in a single year. Compare that to just 1 or 2 out in a field. It's a massive acceleration. And this is where it all comes together. This is the key. When you combine the predictive power of genomic selection with the surgical precision of CRISPR and you add the insane acceleration of speed breeding, you get this incredible formula. This synergy right here is modern plant breeding. It lets us make progress at a speed that would have sounded like science fiction just a couple of decades ago. So now that scientists have this amazing new toolkit, they're not just making small improvements to the crops we already have. No, they are starting to design the crops of tomorrow from the ground up. Crops built specifically for the challenges of a whole new era. So let's talk about one of the holy grails here, perennial crops. Think about it. Most of our food, wheat, rice, corn, they're all annuals. You have to plow the field and plant them every single year, which just destroys soil health over time. Perennials are a whole different beast. You plant them once and they live for years. They build these deep root systems that create healthy soil, stop erosion, and pull carbon out of the atmosphere. The dream is to give our most important food crops that perennial superpower. And this isn't just some fantasy, it's actually happening. Take perennial rice. The results from the first real-world trials are just incredible. This research shows that farmers growing it are seeing their labor cut by almost 60% and their costs drop by nearly 50%. For a small farmer anywhere in the world, those aren't just small improvements. That's life-changing. Now, if you want a truly mind-bending idea, check this out. It's all about learning from evolution itself. You see, over billions of years, organisms have sometimes swapped genes across species. It's a natural process called horizontal gene transfer. Scientists are now thinking, hey, what if we could use CRISPR to copy that process? The goal is to borrow super resilient genes from microbes that live in extreme places and just give those powers directly to our crops. Let's look at how that would actually work. The basic idea is to find a microbe that's an absolute master of survival, something that lives in a volcano or a salt flat, and figure out which genes make it so tough. Then, using CRISPR, you could take those genes that are already perfectly adapted for things like heat, drought, or salt, and stitch them right into a crop's genome. You're basically giving our plants an evolutionary toolkit that took other organisms millions of years to build. You know, when you step back and look at all this, you realize we're in the middle of a really profound shift. For 10,000 years, we just picked the best of whatever nature happened to throw our way. Now, with these incredible tools, we're learning to use nature's own building blocks, its own code, its own evolutionary tricks, to literally design the future of food. And it leaves you with this really big question. Are we moving past just improving nature? And are we now on the verge of actually designing with it?